Okay, I think we're good to go.
All right.
So Nick is going to, we have two speakers today, Nick Mancuso and Eric Kawaguchi, and they're both going to give short presentations. And it looks like Nick's is up already, so why don't you just go ahead and take it away.
Sure. Thanks, Duncan. So everyone can see the screen, right? It's full screen, not like
Yep.
the. Okay, great. So awesome. So what I'm going to talk about today is some of our preliminary findings on trying to identify genes that we believe to be underlying risk for severe COVID-19 outcomes. And so here, what I mean by severe outcomes are essentially hospitalization, right? So individuals who had tested positive, not only tested positive for COVID-19, but were hospitalized due to the resulting symptoms. And so this was a massive collaborative effort. I'll get into more details on the collaboration in a bit. But I do want to say, like, this is all, still somewhat preliminary. We're in the process of writing this up. And this work was presented yesterday at ASHG during like the plenary talk for late-breaking COVID research. These slides are all based off that presentation. So how do I move forward? So just a bit of background, how do we do this analysis? So, well, first, a bit about COVID-19. So SARS-CoV-2, obviously has resulted in an unprecedented spread of COVID-19. And as a result, one thing that many researchers and clinicians have noticed is that individuals who have been infected have there's a wide variability in terms of symptoms and how they manifest across individuals. And so there's been a ton of effort gone into characterizing sort of the genetics of the virus itself, but there's less understanding in terms of understanding how host genetics play a role in terms of this variability. And not only host genetics, but also, importantly, really what we're interested in are the underlying molecular mechanisms, right, that contribute to this variability. And so there are several consortium that have been aggregating genetic data along with phenotype information. And one such consortium is COVID-19 Host Genetics Initiative. And so at the time of the study about the results that I'll be presenting today, there were roughly 3,000 what we call cases. And so cases in this sense aren't really just individuals who have COVID-19, but individuals who are hospitalized due to COVID versus the population. And here population means individuals who have not stated that they have had COVID, or individuals who have been tested positive for COVID, but didn't have any severe symptoms, resulting in them needing to go to the hospital. And so GWIS was done across a bunch of different cohorts, and this was collectively meta-analyzed under this COVID-19 Host Genetics Initiative. And so for this study that we'll be focusing on today, it's about 3,000 what we call cases, and then almost 900,000 controls. So we have extremely well-powered GWAS, right? And at the time of this release, or the freeze that underlie these results, there's essentially a single hit, and it was on chromosome 3. I'll dive a bit into that. So one of the things we wanted to do here is, given these results.
Hey, Nick, where are the controls from?
So, right, so these it's a meta analysis across tons of cohorts. So that it includes UK Biobank, includes FinGen, it includes, at this point, it includes like Million Veterans Program. There are individuals from 23andMe, Ancestry. There's another one called Genomics. I can't remember. There's another sort of European-based consortium that's been jointly analyzed with all this. But at this point, it's, I want to say, upwards of 100 studies that have been meta-analyzed together.
And so what's your feeling about the ability of genomic control methods to deal with that kind of heterogeneity?
Yeah, so no, that's a good question. And so, so there's a lot of heterogeneity, but there's a lot of room for heterogeneity here, right? One in terms of phenotype. And so, the phenotype that we're focusing on is hospitalization. There were several other phenotypes that were investigated, namely COVID infection versus population, right? Individuals who have COVID versus the population, severe versus non-severe, and a couple other phenotype definitions. We focused on hospitalization due to, in my opinion, being the cleanest GWAS signal and probably the best powered to identify anything. My concerns with just individuals who got COVID versus the population is going to be, there's probably true signal there, but it's going to be very noisy because that's just, going to track so much with other factors about, who can be exposed and whatnot. Whereas, focusing on a severe phenotype outcome that removes some of that confounding due to just like being in the wrong place in the wrong time, right, to be infected. And then in terms of heterogeneity across
is the HGI also making comparisons amongst the COVID population in terms of severe versus mild or dead versus surviving?
Yeah, those phenotype results are available. And so, the severe versus non-severe, I think is, something like, it's in the thousands versus like 3,000, right? So it's going to be much lower powered in terms of sample size. And so I think that's why we focus on what they call the B2 phenotype here. And then, as always, it's a meta-analysis. So there's heterogeneity statistics that come along at each SNP. And so things look actually, in terms of the genetic signal, look fairly stable. So my understanding is, or my, as far as my opinion goes, I'm not sort of concerned about there being heterogeneity across studies. But, as always, like population structure and stuff like that are always things that we need to be concerned about. But at this point in time, I'm not super convinced that that's a confounding, playing any sort of role in confounding. Great. So this consortium made these results publicly available as they're being generated at each freeze. And so what we wanted to do was to see if we can identify the susceptibility genes that drive this genetic risk factors, right? And so, this is GWAS, as with all, it's a complex phenotype. As with all other our GWAS on complex phenotype, anytime we find a hit, it's almost always going to be non-coding, right? So it's challenging to identify what the proximal target gene is. And so our job was to try to prioritize genes by taking an integrative approach. And so our main questions were, do total gene expression levels correlate with hospitalized outcomes? And so to do that, we used the transcriptome-wide association study using the latest results from GTEx. For any of the genes that we may identify, we wanted to see if there was also evidence of genetic regulation that's partially independent from EQTLs, right? Just to see if there's, if we prioritize a gene, it's great that it's not sort of surprising that it's going to have an EQTL signal, but we would like to say is there additional evidence of genetic control? And so we looked at that through ASE. Similarly, rather than total gene expression levels, we looked at do individual splicing variants correlate with hospitalized outcomes. And so here a splicing variant is not capturing the total abundance of a particular gene expression, but all the transcripts that are produced, what proportion carry a certain, what have spliced out a particular exon. And then, this is all well and good in that we can generate this giant correlation table, but we wanted to validate our findings through some way. And what we did was we leveraged EHR records in the Vanderbilt Biobank, BioVU, and performed 2 large scans with predicted expression, a PheWAS and a LabWAS. So I'll get into detail about what those are a bit later. And then, as always, interrupt at any point if anyone has any questions or needs if I should clarify anything. So just a bit of background. What is a TWAS? So transcriptome-wide association study leverages population data of gene expression and genotype. It builds predictors of gene expression based on genotype that are local to the gene. It relies on mostly healthy individuals. And the reason why we believe or we have confidence that this approach could work with complex diseases or quantitative traits is that despite healthy individuals underlying the reference panel, risk is going to be spread out across the genome, and healthy individuals will carry risk alleles, and therefore that, despite never sort of manifesting any disease, therefore, it's a good approximation to build predictors in those data sets. And importantly, I think like the key feature of GWIS is once these predictive models have been built, we don't need individual level genotype data to do the prediction. We can take the summary statistics from GWIS directly and apply our weights to that to get a score in the summary level space. And so the sufficient statistics there are really just the marginal correlations and some estimate of the correlation structure. And again, we can leverage things like 1,000 genomes to get at that. So we use all the tissues in GTEx V8 to take an unbiased approach. We use total expression and also alternative splicing. And so, what GWIS gives us is really two things. The first is that it gives us kind of a mechanistic hypothesis, right? We get an association signal now for the expression or alternative splicing of an individual gene or isoform, which is great because that provides a list that we can sort of functionally follow up with. But in addition to that, we're testing far fewer hypotheses, right? In a GWIS, we test millions of SNPs for association, whereas in a TWAS or a splicing TWAS or any of these integrative approaches, we're often testing tens of thousands of hypotheses. So we have reduced testing burden, and what that enables us to do is essentially find novel risk regions, right? And I'll sort of delineate between these two sort of lenses later on. Okay, so that's all well and good. That's kind of the setup. So we applied this approach and we aggregated the results across tissues. And what we ended up finding were two risk regions. One at chromosome 3, where we saw that GWAS peak earlier, and another on chromosome 21. So at chromosome 3, there are a bunch of immune-related genes that we identified, CCR9, SLC6A20, for instance. Many of them have some involvement in either cytokine regulation, chemokine regulation, or are known to be involved in some inflammation-related pathways. So it's starting to fit with like this consistent notion that there's dysregulation of the immune response in some way that leads to these like sort of varied severity in patients. And so similarly on chromosome 21, IFNAR2 is involved in viral immunity. So again, it's sort of fitting with this concept of we think that there's dysregulation at these genes that are involved in some role in immunity or immune response. Now, so of these genes, we wanted to follow up with any additional evidence of genetic control of these loci. And to do that, we looked at allele-specific expression. And so what this captures is really, rather than EQTLs or these predictive models in TWAS are really just fitting a linear model to some normalized expression values and seeing if sort of we have signal there. Allele imbalance is another sort of independent way to capture evidence of genetic control. And what you can do is if you have RNA-seq data for individuals who are heterozygous at any SNP, right? So it means that on their two parental haplotypes, at a given site, they have different alleles. We can see if there's an imbalance on the amount of reads that map to one haplotype versus the other, right? And if there's an imbalance, what that means is that maybe it's not necessarily that allele that they're heterozygous for, but it's tagging something nearby that's altering the amount of expression produced for that gene on that haplotype. And so SLC6A20 showed evidence of allelic imbalance, so further supporting the hypothesis that expression of this gene has genetic control of this locus, and that's responsible or consistent with explaining the risk at this locus. Okay, so all the results so far were using models of total gene expression. The next thing we wanted to do is see if splicing, individual splice variants tracked with COVID hospitalization at all. And so we performed a splicing TWAS, again, using GTX-V8. We identified 4 genes across 46 tissues. Of these 4 genes, we identified 4 splice junctions in total, right? So there can be multiple alternative splicing variants that map to an individual gene. Of all of these, I think the only unique gene that we found through this approach that was not identified through TOS was XCR1. And again, we also recapitulated like IFNAR2 and UN21. So something else that we wanted to look at was, aside from expression, do we see if methylation levels at these loci track with disease risk at all, right? So we've kind of built this model up that expression or alternative splice variants of these genes or a subset of these genes are responsible for risk, but we still have less understanding of individual mechanism, right? Like how are genetics regulating these genes, and how does that alter risk? And so one hypothesis could be that there's varied methylation, that a SNP disrupts something that influences how methylated CPGs can occur. And so, at chromosome 3, there is sort of a really strong signal for CPGs that fall within this gene LZ-TFL1. And so we combined the signals in the GWAS with CPG data, expression of this gene, and what we found was, sort of unsurprisingly, that yes, we have variants at this locus that contribute to risk, right? We're focusing on GWAS, so that's not surprising. But we did see that these variants track with differential methylation at alleles that fall within this gene. Those CPGs also track with the expression of this gene. However, when we try to do predicted expression based on these CPGs, we don't see a signal. So I think there's still a little bit more work that needs to be done here to sort of disentangle how methylation is playing a role in risk at this region. So earlier I mentioned that TWAS and all these integrative approaches provide mechanistic hypotheses, right? Like things that we can identify and go to the lab and functionally validate. But the additional benefit is that they have better power to identify just genomic risk regions on their own, right? So GWAS requires a p-value threshold of 5 × 10⁻⁸, whereas TWAS is much, much lower. And so in our data at the time, we had identified a hit on chromosome 21. And if that is a trick, sample size increases, we should see GWAS signal, right? And so more recently, the latest release essentially doubled the number of patients from size of the we found that there's strong GWAS signals as identified novel risk regions seems to be well supported. It's proper context. It's clear that just picking any number of those genes, that there's some involvement in immune function, but we need some way to validate them. And so what we did was we predicted gene expression into roughly 70,000 patients of European ancestry that also have EHR or medical records in the Vanderbilt Biobank BioVU system. And what we found was we had associations with IFNAR2, with bundle branch block, and migraine symptoms. And these are both consistent with symptoms observed in COVID-19 patients where we have heart arrhythmias or varied neurological responses and migraine for patients, right? So this is still, again, providing some additional support that the genes that we're finding and the phenotypes they affect are consistent with what's observed in COVID patients. And so similarly, not only do we look at, what clinical phenotypes may be related to the expression of these genes, we wanted to see what sort of biomarkers or labs are associated. And so using the predicted expression in the biobank, we also tracked what they're correlated with in terms of laboratory tests, right? And so what we found were essentially additional markers of inflammation, right? So several of these genes had strong hits for basophils, monocytes, which are essentially reflect varied white blood cells, as well as Ig clearance, which is important for IL-10RB, which is right near IFNR2. And so the takeaway now is that, okay, we found these genes, we looked in the BioView, we see that they track with clinical symptoms that are consistent with what's observed in COVID patients, and in addition to that, we see that they track with laboratory tests that are consistent with markers of inflammation. And so it's, further supporting this hypothesis that, it's a dysregulation of the immune response across patients that's leading some individuals to have these severe outcomes versus those who don't. And so the conclusion here is that using roughly 3,000, a GWAS of 3,000 hospitalized patients due to COVID versus approximately 900,000 who don't, we identified 6 genes at 2 loci. Of these genes, SLC6A20 had additional support of alleles of genetic control through ASE. We found 4 genes through splicing, and they were at the same loci. And so I don't think the story here is that all these genes and all these splice variants are driving risk. We'll need to do some additional follow-up to sort of prioritize really what's going on here. IFNR2 locus was validated in the larger GWAS, right? Our model predicted that we should see GWAS signal there as sample size increases, and that was exactly the case when the sample size doubled. And then again, our PheWAS and LabWAS in the BioView provided clinically relevant outcomes and tests that are consistent with immune response. So there's a new result, so our upcoming work focused on the latest freeze. And so even this slide that I'm showing here is actually outdated as of this point because there's been an additional follow-up release. And so we went from having 6 genes at 2 loci now to have approximately 20 genes across, I think, like 10 loci, which this figure doesn't necessarily reflect. And of those genes, several more are involved in immune-related function. And sort of interestingly enough here, we see a gene DPP9, which is known to have a strong role in like pulmonary fibrosis, right? Which is this lung disease that affects like scarring tissue, and that should have again is like consistent with what we expect to see in individuals that have sort of severe outcomes. So this wasn't solely my effort. This was the joint effort of a lot of groups, and really this was made possible by the COVID-19 HGI consortium, right? They're the ones that did the legwork, that built this massive consortium to aggregate the GWAS across all of these individual sites. And so we built a TWAS working group that aims to identify the sort of the mechanisms and genes underlying these observed genetic risk factors. And so that's in collaboration with Renato Palamonte. His postdoc Gita did a lot of the TWAS efforts here and put together these slides and gave a fantastic talk yesterday on ASHG. Leah Davis's lab at Vanderbilt. So Kritika Singh and team did the LabWAS and PheWAS. So they've again been like monumental in driving these validations. Several members from my group, Shami and Zayun, did a lot of work in building some of these weights and doing some statistical follow-up, as well as like Bogdan's group at UCLA and several others like Nava, who's in Pej's lab, did the ASE, as well as Loic, who's been looking into methylation. And as well as others. I think there are some folks that we're still trying to expand efforts here, and trying to integrate a couple additional results last minute to provide more functional validation for our findings. And so we're in the process of writing this up. I think it's already long past due to have been written up, considering we presented on it now. But we're hoping to get a manuscript or preprint out, in the next coming couple weeks, and hopefully have it under review soon. So happy to take any questions that anyone may have.
Well, I'm blown away by the complexity of the work that you've done. I just wish I had heard this before yesterday when I had to sit through a day-long study section on the subject. So
Oh,
And
well,
being
thank you.
relatively ignorant myself on a lot of this stuff.
Yeah, I mean, everything's just moving so fast. It's hard to keep up just with like with all of our other duties. It seems like every week there's a new series of papers that come out. And so it's been tricky. But again, a lot of the trainees who are driving these analyses have been just awesome at really pushing things out as new data come in. So it's been a really fun collaboration. And we're still open. The group is open. We're still, if anyone is interested or wants to be involved, please contact me or anyone else in the group. We're definitely willing to expand these efforts. And we're not sort of envisioning just this single paper and that we're done and hands off. I think there's still a lot of other analyses that we could look at in terms of how responses differ by ancestry. Do we see replication in non-European individuals? That would be really important to kind of provide additional support for our findings.
Are you getting
So Nick,
involved in the various USC working groups on COVID?
Ooh, two questions. Who wants to go first?
Go ahead, Duncan.
I was asking if you're involved in the various PM and wider USC working groups on COVID.
No, I'm not. And I would be happy to chat with folks involved here. Like I said, we haven't done any functional validation yet. We're still working out the details on where that might occur. But again, nothing's been decided. So I'd be happy to chat with anyone who's capable of doing any kind of functional validation in any sort of cells that would be relevant here.
Well, I would start by having a chat with David and Frank, who are leading the preventive medicine working groups.
Okay, yeah, we'll do.
Tim.
I was just going to ask, the early publicity, the press picked up the ABO blood group as being associated with risk. Is there anything that you got that wasn't one of your results?
So at the time of this, what we submitted to ASHG, it was not. However, in the latest release, it is. So ABO has strong signal. And so in addition to GTEx and predicted expression, we've now also incorporated predicted protein abundance. So rather than just the RNA levels, we also build predictors using this study that has like plasma proteins, circulating plasma proteins. And so ABO is a really strong hit there. And which is interesting because I think, the first couple papers that said ABO, honestly, I was a little bit skeptical because it sounded like their study design may have been confounded with based on how they got participants in the first place. But, it seems to be.
But it was in 3 different continents, I think. So
yeah. So yeah, exactly. So, I mean, it seems to be pretty well supported at this point, which is interesting. And so, again, it seems like ABO has a long history of having a role in protective effects, or, deleterious, depending how you look at it, for viral infections. So it makes sense. And so again, it's fitting with this broader pattern of, we're seeing either dysregulated immune function or something involved in like lung phenotypes or just defense in the first place.
And any other questions for Nick at this point? So,
cool.
Oh, someone say something?
Thank you.
No, I was just saying cool. Thank you.
All right. So,
Thanks.
Eric, I've just unmuted you. Do you have slides you want to share?
Yeah. Let me just bring it up. Can everybody see the slides up here?
Yep.
Okay, great. In hindsight, I should have been before Nick. I think Nick's talk was a lot more interesting than what I'm going to be giving, so it would have been a better way to end the seminar series. But it is what it is, right? So today I'll be presenting some work I've been doing. It's joint work with JP and Jim over here at USC. This is sort of a quick introduction to what I'm doing. It's genome-wide interaction scans for right-censored time-to-event data. So just, as a quick recap, in a GWIS, focuses typically on assessing marginal associations of each SNP with a particular trait. but some SNPs may not be identified in GWAS due to having weak marginal effects, consequently low power. But it's possible that these SNPs may be quite important in subgroups of the population, where these subgroups can be defined by an exposure. So this could be like, you know, like a demographic characteristic, maybe an environmental exposure, maybe even like a treatment group. So it leads us to this following question, which is, by using a genome-wide interaction scan, can we find new genes that were not found in the primary GWAS scan? You know, apparently from my talk, it's going to be focused on right-censored data. But before we dive into that, I just want to kind of go over the more classical scenario in terms of a case control study. So if you consider the following case control study, where we have n subjects, n naught is going to be the number of controls, n1 would be the number of cases. DI is going to be the disease status for the n subjects. It's going to take on, you know, values 0, 1, 0 being controls, 1 being cases. EI will be defined as an exposure status, and I put exposure in quotations because this is a very broad term. You could think of this as environmental exposures. If you're looking at a treatment, you could look at this as, you know, the treatment indicator, and so forth. And then we have GIJ, which is going to denote the jth SNP. And GIJ could take on, again, a number of different types of values. Without loss of generality, we're going to assume that both E and G are going to be binary. And you could view this in the following case. E is a treatment indicator, 0 being placebo, 1 being a treatment. And then GIJ equaling 1 would just imply that we are genotyping this as AA, or uppercase A, or uppercase A, lowercase a. You could look at this as an additive model or a recessive model as well, but just for the purposes of this talk, we're looking at it as being a binary variable. So a standard GWIS, or genome-wide interaction scan, will just allow you to just test each SNP in turn for a multiplicative GYE interaction. Since D is going to be, since D is a binary variable, this is viewed as a logistic regression. And what we're focused on here is looking at the following term over here. This is gamma G by E J. This is sort of like the interaction effect. And we could test these M hypotheses, whether or not this effect is 0. Since we're looking at M different hypotheses, we would need to adjust from a multiplicity to control the type 1 error rate. For example, we could use a typical Bonferroni correction where alpha star is going to be alpha over capital M. When M is in the thousands, tens of thousands, even millions, like we have in typical GWAS, this leads to very conservative values of alpha star and can result in very low power. So we don't really solve the issue that we were looking at before, just using a standard GWIS scan. So what's been proposed in the literature is this idea of two-step approaches. And so this provides greater power than the standard scan being what I presented above, while retaining type 1 error. The way it works is as follows. Step 1, we're going to filter or order SNPs based on some sort of independent screening statistic, and I'll talk about this later. And then step 2 is we're going to test the same null hypothesis looking at the treatment effect at a modified significance level, where the significance level is going to be based on the orderings from step 1. So just to give you an example, you know, there's a lot of things here. What's step 1? What's the screening statistics? I'm going to go over this one by one. So in terms of screenings, there's 2 well-appreciated ways to perform step 1 screening. The first one is a subset testing approach by Cooperberg and LeBlanc in 2008. This one's pretty straightforward. So we basically test each of these M screening statistics to a pre-specified significance threshold that I'm going to call alpha naught. And then we're going to test the M interaction terms, lowercase m interaction terms at a significance level of alpha over lowercase m, where m, lowercase m, is the number of SNPs that sort of passed this first initial screening round that was dictated by alpha naught. So this could be, you know, you could look at this, it's subsetting the approach, because even though capital M can be large, lowercase m can be rather small. You know, for example, it could be maybe instead of having to test 10,000 interaction terms, you're only testing 50 of them. So instead of dividing your alpha by by 10,000, you're just dividing it by 50. So you're having a less conservative type 1 error. And so it's sort of subset testing. It's kind of dichotomous. You're either in the testing group or not in the testing group based on alpha naught. So a more relaxed approach is this idea of a weighted Bonferroni. by Ionita-Lodson in 2007. This is sort of in between a subset testing and a classical Bonferroni scenario. So what we're going to first do is rank these capital M screening statistics in decreasing order, and then we're going to suppose an initial bin size. For this example, I'm going to just say, let's say 5. So what's going to happen is that the first 5 interaction terms that correspond to the 5 largest screening statistics, those interaction terms are going to be tested at a significance level of alpha over 2 over 5. And then the next 10 of them, so SNP 6 to SNP 15, are going to be tested at a significance level of alpha over 4 over 10. And then the next 20 below that are going to be tested at alpha over 8 over 20, and so on and so forth. It's just going to keep on decreasing. The type 1 error, or the significance threshold, will keep on decreasing within these bin sizes. Both approaches, the subset and the weighted Bonferroni, ensure that the family-wise error rate is retained at alpha. I know this is a lot to digest, and so the best way to sort of view this is as an illustrative example. I think this way people can understand what's going on. So we're going to suppose we have 100 SNPs, and we'll let chi-squared j be the step 1 screening statistic. I didn't talk about how we're going to derive it, but this is say chi-squared j is going to be these step 1 statistics for each SNP. And we're just trying to find the significance threshold alpha star for the interaction term they want to test with while controlling the family-wise error rate at alpha equals 0.05. For a classical Bonferroni, alpha star is just going to be, in this case, alpha over 100. But we want to see how this value is compared to what you would get with the weighted test or the subset approach. So here are the 100 screening statistics unordered. The first thing you want to do is order them in decreasing order. So you can see that the largest chi-squared statistic is 10.3, then it's 9.2, 7.8, so on and so forth. And when we get to the lowest one, it's pretty much 0. And so this is how the alpha star is going to look for each interaction test. So with subset testing, I'm saying here that alpha naught, that initial alpha that we had before, is going to be 0.05. And when you look at an alpha of 0.05, only the top, based off of these chi-square statistics, only the top 8 are going to be kept in the subset, which means that we're going to divide alpha by 8. So we're going to test each interaction term. So the interaction term corresponding to the 64th SNP is going to be tested at a significance level of 0.0063. Same thing for the interaction term corresponding to the 18th SNP, and so on and so forth. And anything that didn't pass the initial threshold will not be tested, or you could view it as essentially being tested at a alpha of 0. When you look at the weighted Bonferroni test, and for the weight, I'm going to be considering it to be 2, just so it's easy to see in this small table. We can see that the first 2 SNPs are going to be, so SNP 64 and 18, their interaction term is going to be tested at a threshold of 0.0125. The next 4 are going to be tested at 0.0033, and then the next 8 are going to be tested at 0.0008, and then the last, bin they're going to be tested at 0.0001, essentially 0. And then we can see here the actual classical Bonferroni. So this is just alpha over 100, and we can see sort of the benefit of this, right? Because, well, this sort of, the alpha spending is the same across all SNPs. The alpha spendings for subset and weight are going to be much more liberal in terms of the SNPs that have a larger marginal association or a larger step 1 statistic than those that have a worse step 1 statistic. So, the question is, well, can we get an informative step 1 statistic so that the interaction can be tested at a more relaxed alpha level than alpha over M? So this is sort of the way you're doing subset, or this is sort of the way you're doing step 1 and step 2 screening. Now the next question is, well, what should we use as our possible screening statistics? And there's quite a few that have been proposed in the literature. This is, by all means, not an exhaustive list. This is just 3 simple ones that I feel like I could just talk about here. So the first one is by Kuperberg and LeBlanc, which is what I'm going to call the marginal G by G by E test. So the screening statistic here is going to just look at the univariate effect of the SNP on the disease. So we're not taking into account the exposure or the interaction term. And then the screening statistic is just going to be looking at the wall test squared, which is going to follow a chi-squared 1 distribution. So this is this MG by G by E test. Another one that's been proposed by Mercury et al. In 2009 was this G star by GWIS, which instead of looking at the outcome, you're looking at the exposure, if the exposure is binary. And so you're looking at the probability of being exposed given the gene and given the SNP. And then the same thing here, this walled statistic here corresponding to this is going to be distributed chi-squared 1. And then lastly, Gellerman et al. Sort of came up with a 2-degree freedom test that sort of adds these two contributions from this M, G, and G star test. And basically is saying you use a screening statistic that's the sum of the two. So the sum of T beta GJ and T delta GJ, that comes at the chi-squared 2. And they show that of all three methods, this one sort of has the highest power in terms of this two-step approach. And so that's sort of the state of the art, what sort of has been happening in terms of case control setting. So now the motivating question is, well, does this hold for right-censored survival data? So Jim sort of came up to me and we kind of talked about doing this. It seemed like a natural extension is to do this for time-to-event data. And at first I was like, okay, this seems like a very interesting problem, a very simple problem. You just do the same thing and copy and paste it, except you just replace the model with a Cox model, for example. It turns out that that's not actually right, and I'll show why here. So we're going to first consider a randomized controlled study. Say here we have n subjects, T tilde is the observed survival time, where Ti and Ci are the true survival and censoring times respectively. Delta I is going to be a censoring indicator, indicating that the observed time is the true survival time. Again, that E being exposure, the G be the SNP variant. We're going to, again, without loss of generality, assume that they're binary, and then we're going to assume the usual right-censored survival data assumption. So for example, we assume that T and C are conditionally independent given E and G and any other confounders in the model. So we're going to assume, if you assume a proportional hazard assumption, you know, we're just basically looking at the following model. This looks very similar to the first model we looked at, except instead of looking at the logit, we're looking at the hazard. So HT given G, if so, you know, you have this conditional Cox model where HT given X is the conditional hazard function at time T given X, then H naught of T is an unspecified baseline hazard function. And so this is a standard Cox GWIS. Again, this doesn't avoid any problems. We're still going to be severely underpowered for large M. And so, you know, can we adopt these two-step approaches and do this for the Cox model? So, you know, we can parallel Kuperberg and LeBlanc's step 1 screening approach. Remember, that one was looking at just the marginal effect of G on the case control status. So in this case, we're looking at the marginal effect of G on survival. So this is looking at a marginal Cox model. And then the question that we have here, in order to make these two-step screening tests valid, is that beta G and gamma G by E have to be asymptotically independent. That's sort of a very important and crucial assumption in order for these two-step tests to hold, to retain type 1 error. And Dai et al. Proved this result for GLMs under a canonical link. So the case control example with the logistic regression, that works out, everything's fine. They showed that theoretically, they showed that their simulations. And you know, it was what you'd expected. And then the question is, well, can this hold for the Cox model? And to the best of my knowledge, a general result for the Cox model has yet to be shown. But we conjecture that in general, this is not true. And so one reason why is this issue of bias due to omitting covariates. This is an issue with the Cox model. So it's well known that the estimates are biased towards 0 if important covariates are omitted from the model. This is a very simple toy example that I'm presenting. So here we see that beta 1 and beta 2, I'm going to just say that the true effects are 1. We can see that if we look at the full model, where we include both x1 and x2, that the estimates are very close to 1. And as n increases, you know, it's the same. You expect it to be unbiased. But what happened if we were to look at the model where we only include x1 and we forget x2, or we only include x2 and forget x1? So in the case of the marginal test, this is sort of what you're doing. And we can see that the estimates are actually a lot or biased downwards towards 0 for both these cases. And mind you, this happens both when you have both positive effects or one positive, one negative effect. The censoring rate here was about 95, or the event rate was about 95%. So censoring rate was about 5%. And so this is looking at the model with almost having the entire cohort having the event observed. And even then, the bias doesn't go away as n increases. So we have this bias towards 0 that would happen if important covariates are omitted from the model. This isn't only unique for the Cox model. In fact, for the logistic regression, you have a very similar situation where the estimates are sort of biased towards 0, except the bias there isn't as severe as it is for the Cox model. And that's probably due to the fact that for the Cox model, since we're looking at the survival time, not omitting an important covariate is sort of affecting this pH assumption that you have. So this is sort of a phenomenon that's been well known. It's sort of the basis and motivation for frailty modeling under unobserved heterogeneity, except in this case, we have observed heterogeneity because we know what's actually driving this. In this case, it's going to be the exposure or the genetic variant that we're omitting. So I propose to look at a modified step 1 screening statistic. So instead of using just the marginal effect of the survival time on the SNP, I'm just also including the exposure. This is nothing fancy. It's just a main effects Cox model. And now the question here is, okay, well, same thing, is omega G and gamma G, are they going to be asymptotically independent? Or gamma G by E, are they going to be asymptotically independent? And you could show theoretically that they are under specific assumptions. So here we're going to assume that the interaction model is correctly specified. Then under the null hypothesis that gamma G by E is 0, the MPLE for omega hat G and gamma hat G are going to be asymptotically independent. And this only holds for the main effects model. It's yet to be proven that this holds for the univariate model with only the SNP. And so using this, I kind of performed some simulations to show how well this works. So the first thing we're going to look at is the correlation between the step 1 and step 2 estimators. And so the true survival times are generated from the following exponential model with a baseline hazard of 0.01. You assume here that gamma G is 0, so that the only effect that we have is going to be through the exposure or through the interaction term. X1 and X2 are simulated from a bivariate normal distribution, but then we dichotomize X2 via X2. If it's less than 0, it's going to be 0. If it's greater than 0, it's going to be 1. So again, with the bivariate normal assumption, it'll roughly correspond to about 50% zeros, 50% ones, sort of what you would see for a randomized controlled trial. Centering times are simulated from an exponential model with rate 0.05, and that corresponds to an event rate between 60 to 70%. And what we're looking at is the parameter estimates from the following models. So again, the univariate model, where we look at just X1, or in this case, the SNP, and then the main effects model, where we look at the SNP and the exposure. And what we like to see here is you want to look at the correlation between gamma hat G by E from the interaction model and beta hat G, and then the correlation between gamma hat G by E and omega hat G. And we look at this through several different scenarios as n increases. So these are the terms of the correlation. And this is the terms of gamma E and gamma G by E and their effects. So the hazard ratios would be in here. So this will correspond to a hazard ratio of 1 for both gamma E and gamma G by E. So this is a null model. Gamma G, gamma E, and gamma G by E are all 0. And we can see here that, as you'd expect, they both perform almost identically. They're both pretty much the same model, under the assumption that the effects are all null, and that the correlations are rather close to 0. And we can see that here. What's interesting though is when we look at the case where an exposure is present, that the correlation still gets close to 0 for the main effects model. But when we look at the univariate model, that the correlation is not negligible. Still, it's about negative 0.14, negative 0.12. And then when we look at it on the reverse end, when we have a protective effect of the exposure, and so if this is a treatment, this will correspond to the treatment having a positive effect on survival. Again, omega G, the correlation is close to 0, which is what you'd expect. You want these two test statistics to be asymptotic independent, but we can see here that the effect for the univariate model still has a non-negligible correlation. And this is prevalent all throughout the simulation that we have here. I tried this looking at different censoring rates. I tried this looking at different values of gamma G. I also looked at this with different ranges of correlations between X1 and X2, and we see very similar trends. When an exposure is present, we still retain, asymptotic independence for omega G and gamma G by E, but we don't see that for beta G and gamma G by E. And so what this shows you is that the step 1 and step 2 assumption that we have, which is that the step 1 statistic and step 2 statistic have to be independent in order for this to be a valid way to increase power for GWIS, it seems like that wouldn't hold for the marginal G model, but it can hold for the main effects model. And so we want to now assess type 1 error in power. So here we look at 1,000 replicate data sets where N is 1,000, M is 10,000, where M is the number of SNPs. So we here assigned one SNP to be the disease susceptibility locus, so it has a true G by E effect. The minor allele frequency here is 0.3, which corresponds to about 51% carriers under the dominant model. So what this means is that the G variable here is going to be 0, 1, and it has a 50% chance of being 0 or 1. The exposure here is going to be simulated from a Bernoulli distribution with probability 0.5. So this is, again, like a treatment variable. You're either a treatment group or placebo group. Seurat times are generated from an exponential model, the same interaction model you saw before, with baseline hazard 0.01. And then centering times are generated from an exponential model with rate 0.005. And then for the remaining M-1 non-DSL loci, they sort of varied their allele frequencies from 0.1 to 0.25 to 0.4, just to give just different frequencies of zeros and ones all throughout the M-1 variables. So we compare the following approaches. So the standard GWIS approach. So we just look at the interaction term. We test this at a Bonferroni corrected p-value of 0.05 over capital M, which is 10,000. So we would assume that this has pretty low power. We look at the marginal G association step for step 1 screening. So this was looking at the Cox model where we only look at the SNP. Then we look at what I would call the conditional G association for step 1 screening. And this is looking at the main effects model and only looking at the effect of omega G. So that's sort of what we were looking at, which we expect to perform quite well. And then we also, just to throw it in there, we looked at this marginal G plus G star by G by E test. This is the one that we saw before it was proposed by Gatterman et al. In 2012, which works quite well for the logistic regression. And we're just going to see if that works for the Cox model. Again, we're using the marginal G test here. So we conjecture that doesn't work that well, but we're going to show empirically whether or not that's true. Just for simplicity in the simulations, I'm looking at a weighted Bonferroni test with B equals 5. And that's used for step 2 hypothesis testing. What you see here really quickly is that under a whole range of simulations, first thing is that the standard approach typically has low power, and in 2-stage approaches typically have better power regardless. And that's sort of what you see all throughout the simulations. In a case where there's no exposure effect, we see that all the type 1 errors are kind of retained around 0.05, which is what you'd expect. And then the power seems to be doing a lot better using the 2-stage versus the 1-stage approach. But we start seeing issues when we have an actual exposure effect. So when the exposure is true, which we do see here, again, we do see that the power is much better for the 2-stage test over the 1-stage test. But again, for the ones looking at the marginal G association, the type 1 errors are inflated. We don't see that inflation happening for the conditional test, but we see that happening for the marginal test, which means that we have to take into account the exposure in order to model that, in order to preserve type 1 error. And so just for conclusions, this is a quick, not thorough discussion, but 2-step GWIS approaches can be applied to the Cox model for right-censored data. So the answer to that question is yes, it can be done. But there are words of caution. The first thing is that conditioning on the exposure is necessary to preserve type 1 error. Dye et al. Showed that that wasn't necessary for the case control model. It just sort of improves power, but it is a necessity when it looks at the Cox model. So some next steps that I wanted to take with this is, number 1, apply this approach to data. So currently I have simulation results, have this across different model and specification, different parameter estimates, different allele frequencies. You all get the same story. That doesn't change. But the next thing I want to do is apply this to data. The second is, our theorem that we proved only holds under the correct model specification and under the null. And so the question is, can we generalize this result to being under model specification? This is something you would see like sort of paralleling Dye et al.'s theorem. We want to find a more sophisticated way to incorporate the exposure. Currently, I'm only looking at a main effect model, and that's sort of just showing that the main effect model is the correct way to model at least the genetic effect. But is there any way we can incorporate exposure to sort of boost power, sort of what Gauderman et al. Showed in their paper for the case control study? We want to generalize this to exposures without the pH assumption. So currently we're assuming that the exposure has a proportional hazards interpretation on the survival. It's quite natural to believe that maybe this isn't true. And so we want to look at this maybe in a stratified Cox model or another way to model the exposure. And then the last thing is we want to extend this theory and application to various time-to-event settings. This could be looked at, say, a parametric AFT model if you want to assume a survival distribution. If you want to assume a parametric distribution on survival in the competing risk setting, we could look at that too. Or we could even look at this under various epidemiological cohort designs, such as a nested case control or a case cohort study. Yeah, so that's currently what I have. And I know it's perfectly 1 o'clock, but if anybody has a few questions, I'll be happy to answer them.
Very interesting. And I know some people have to go to the 610 class, but we can keep this Zoom open for a while if people have other questions.
Hey, Eric, great job. So do you have plans for what type of exposure or what type of GWAS data to apply this to?
Not as of yet. I think I'm still pretty open to either. Currently, the way the methodology is being formed, I think you could look at a continuous exposure or a dichotomous exposure. As of now, obviously, when we look at stratifying and stuff, you would like to have an exposure that is not continuous. But yeah, as of now, I'm open to looking at any sort of time-to-event GWAS data that's available.
Really cool.
Any kind of treatment trial would be fair game for this, and I think that would be a very natural place to do that. And you have the nice feature that if treatment is randomized, you've got independence with exposure right off the bat. We're still trying to figure out whether we can bring in that kind of second screening test, kind of an E versus G kind of screening test. And what Eric didn't show you is a lot of other work that he's done investigating some avenues that way that haven't panned out yet. So I haven't quite given up hope that we can get something that'll work in the survival setting that's akin to this Mercury approach here. But that's still work that's kind of ongoing. Well, I think cohort studies that have time-to-event data, also would be fair game. You just have to be a little careful about, the exposures and the potential biases that can come in if it's not a randomized trial.
What you're calling the G-star test here, the Mercury test, works for case control data because you're oversampling cases relative to controls. That wouldn't be true in a survival context if you're including everybody. So I think the question I wanted to ask is whether or not an analysis that would select, let's say, one control from each risk set for each case, whether that would be valid, whether it would be orthogonal to the stage 2 test and so on, and might buy you some power.
I think that might be interesting to look at.
Yeah, that's one of the ones that you've said you've been exploring.
No, we have not tried that one. So I definitely think that would be worth trying. No question.
So the idea that you're talking about, Duncan, is just sort of doing like a matched case control on the like in terms of the risk set.
Yes, just for the
Okay.
first stage test, using everybody at the second stage.
Yeah, that might be interesting to look at.
You know, you can show that in a clinical trial setting, you know, G and E are going to be independent at baseline due to randomization. But if there's a strong G by E, there's kind of an induced G to E association that happens that's a function of time since randomization, because you've got people selectively dropping out of the risk sets in a way that's biased by their G and E because of the interaction. Of course, if everybody failed by the end of the trial, then you'd be back to a situation where there's no association again. So there's kind of a time-dependent relationship between G and E that so far we haven't really been able to effectively capitalize on. We have a couple, you know, harebrained schemes, but Eric has pretty effectively showed that they're not independent of that step 2 test. And that's where that's the rub. So maybe the approach you suggest, Duncan, may be the way out of it. So it's definitely worth trying.
Yeah. Yeah, I think that like a main issue here in general is that when it comes to Serawa data, you know, the Serawa time is affected both by G and E, and in turn the G by E effect. I was looking at very simple like this slide on omitting covariates that I had here. You know, for logistic regression, it sort of, you know, came out to be still slightly biased, but not as biased. But what was interesting was when I looked at a nested case control sampling, that sort of kind of goes away. Not goes away, but when I looked at a nested case control design, I expected it to be something close to the Cox model, but it was actually something closer to the logistic regression model. So I think there's actually, when you sample, you're sort of breaking this correlation a little bit, which might allow you to have an independent statistic. But I'm assuming if the number of cases you're sampling is close to the number in the risk set, you'll probably still get it to be closer to the Cox model setting, which is sort of another paper I was kind of interested in writing, is showing how that affects it. So if you have nested case control with 1-to-1 sampling, you're all good to go. But with 5-to-1, 10-to-1 sampling or whatnot, you're probably going to be closer towards the Cox model setting than you are the case control logistic regression setting. But that might do it in terms of doing something similar to the Mercury paper.
Any other questions for Eric? Well, I thought this was really a great session with 2 good talks, and very much looking forward to the next one, which will be in 2 weeks, because we have a faculty meeting next week.
Do we have a speaker set up for 2 weeks, Duncan?
We do. David and I'm blanking on
Abigail.
her name. Yeah, are going to talk about COVID.
Oh.
And then the week after that, we have Brahmar coming.
Good. I thought COVID wasn't a problem anymore.
Apparently after November 3rd, it won't be a problem anymore.
Wait till next Tuesday and we'll find out whether we have a problem.
Yes, exactly.
All right. Well, bye, everyone.
Okay.